Hello, and welcome to Oceanbites Out Loud. My name is Diana, and I'm a PhD student at URI GSO. Today, I'll discuss what it's like to do research at sea. Do you wonder what it's like to do oceanographic research aboard a seagoing vessel? What types of research are conducted out at sea? Where do scientists set up their labs? Listen on to hear about my experience aboard the RV Endeavour. At the beginning of February, I spent 5 nights aboard the RV Endeavour with about 15 other scientists from the University of Rhode Island, Woods Hole Oceanographic Institution, and Wellesley College. We participated in the winter transect for the Northeast U.S. Shelf Long-Term Ecological Research Project. What is the NES LTER? Since 1980, the National Science Foundation has funded 28 LTER sites around the U.S. and Antarctica, many of which are terrestrial. LTER sites are important because they are studied over long periods of time. Some of the sites have been researched for over 30 years. Collecting the same measurements over time enables scientists to understand how processes at a particular site may be changing, either from natural variation or from human influence. The NES LTER is a recently funded marine site, examining how planktonic food webs are changing on the northeast U.S. continental shelf. There are 4 NES LTER cruises evenly spaced throughout the year for scientists to study the seasonal and long-term changes of food webs. Phytoplankton are at the bottom of the marine food web, and they fuel the productivity of ocean life. They produce oxygen through photosynthesis. In fact, did you know that every other breath of oxygen you take comes from the ocean? Zooplankton, the next step in the food web, eat phytoplankton and in turn are eaten by various commercially important fish. The Northeast U.S. Shelf is one of the most biologically productive areas in the world. Thus, it is important to understand how physical, chemical, and biological processes affect the connectivity of organisms on the Northeast U.S. Shelf. What does a normal day at sea look like, you might ask? Time on a research vessel is very expensive. Think about all of the costs that go into running a research ship, such as the food we eat or the fuel needed to power the ship. To be as efficient with our valuable time as possible, as a group, we worked 24 hours a day for 5 days straight. Don't worry, each scientist is assigned to a 12-hour shift. My shift was noon to midnight, and by the end of the cruise, I was sleeping for almost the entirety of my off shift. At the beginning of each day, I had my breakfast, which was actually lunch, and figured out where we were on our cruise track. Depending on how close we were to the next station, I got suited up in a warm Mustang suit and headed out on deck. A Mustang suit is a large, insulated orange suit designed to keep you warm and protected in the rare chance you fall overboard. We collected water at multiple depths at each station using a CTD rosette, which measures various water properties such as salinity and temperature. The instrument also contains large bottles called Niskin bottles that the scientists program to close at various depths in the ocean to collect seawater. As I mentioned earlier, phytoplankton fuel the marine food web. Because phytoplankton photosynthesize, they have chlorophyll A, a pigment in their cells to help them absorb sunlight. Using a fluorometer, we measured the fluorescence of the water to determine where in the water column the chlorophyll maximum occurred. The determination of where to sample in the water column, which is basically a piece of the ocean from the surface to the depth, is probably one of the most important factors. Well, besides making sure the bottle's properly closed to collect our water. You also might ask what specific research took place. For my lab's research, we sampled water at 3 depths within the water column. What this means is that for 5 days, I was a glorified Brita filter and couldn't have been happier because I was in the middle of the ocean surrounded by nothing but water. I filtered liters and liters of water for chlorophyll and DNA analyses. Just like you and me, phytoplankton have DNA, the genetic code for how a cell or body functions. Filtering water for DNA gives us the inside look into what is in the water at various depths across all of the stations. Other work on the cruise involved sampling zooplankton and carrying out experiments to determine how quickly the zooplankton ate the phytoplankton, which is known as a grazing experiment. Coupling these multiple analyses together over a long period of time will enable us to better assess marine food webs both now and in the future. When there was a break in the action, I went up to the flybridge, which is the very top of the ship, to appreciate the opportunity I had to work alongside amazing scientists and admire the wonderful world we live in. I'm looking forward to the next cruise. Thanks for listening.
This has been Oceanbites Out Loud. Check out this post and other interesting posts at oceanbites.org.